Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 52. The topic is glycolysis and the citric acid cycle connect to many other metabolic pathways. Discussing agenda for the topic will be glycolysis in the citric acid cycle connect to many other metabolic pathways, the versatility of catabolism, biosynthesis, anabolic pathways, regulation of cellular respiration via feedback mechanism. Lecture outcome. After watching and listening this lecture, students will know how glycolysis and the citric acid cycle are connected to other metabolic pathways. Introduction. In this section, you will learn that glycolysis and the citric acid cycle are major intersections of the cell's catabolic and anabolic pathways. So in this lecture, we will discuss how different metabolic pathways are related with each other. Our focus will be on glycolysis, which is of course the first stage of cellular respiration, and then citric acid cycle or Krebs cycle, which is the second stage of cellular respiration. So our focus in this lecture will be that how the metabolic products or the intermediates of other pathway can become a part of glycolysis and citric acid cycle, and how the intermediates of glycolysis and citric acid cycle can enter into other metabolic pathways. The versatility of catabolism. Throughout this chapter, glucose has been used as an example of a fuel for cellular respiration. So in the majority of the lectures in this chapter, our focus was on the breakdown of cellulose molecule for the generation of ATPs. We discussed glycolysis, Krebs cycle, electron transport chain. But free glucose molecules are not common in the diets of humans and other animals. We obtain most of our calories in the form of fats and proteins. So the starting point of glycolysis is glucose molecule. But if glucose is not available in your diet, then how your system is going to generate energy from other macromolecules like fats and proteins? So carbohydrate such as sucrose and other disaccharides and starch, a polysaccharide. So quite often the free glucose molecules are not available in our diet. We consume complex carbohydrates in the form of disaccharides and polysaccharides. So is there a mechanism in the body which can derive the basic monomers from these complex carbohydrates, or is there any mechanism in the body which can generate energy from other forms of macromolecules? All these organic molecules in food can be used by cellular respiration to make ATP. So fats and proteins can also be used as a source of energy. We will discover how. On the right side of your screen, you can see the catabolism of various molecules from food: proteins, carbohydrates, fats. These are the three classes of biological macromolecules. Proteins are broken down into their building blocks called amino acids. Carbohydrates are broken down into their building blocks, sugars. Fats are very different in structures. They have structural diversity like phospholipids, fatty acids, waxes, etc., cholesterol. So the basic subunits of the fats are fatty acids and glycerol. Now, how these monomers of different macromolecules can become part of this channel, this process of glycolysis and citric acid cycle. So you can see here amino acids can be broken down and acetyl coenzyme A can be produced from amino acids and then acetyl coenzyme A can enter into the citric acid cycle. Similarly, acetyl coenzyme A can also enter another route and they can also leads to the formation of amino acids. But remember that in normal circumstances, amino acids or the proteins does not provide energy. If the reserves of carbohydrates and fats are available, then proteins in such circumstances does not provide energy. But when the cell runs short of its carbohydrate and fat reserves, then proteins are used as a source of energy and the amino acids can be broken down and acetyl coenzyme A is released, which enters into Krebs cycle. Now sugars can directly enter glycolysis. Remember, the starting point of glycolysis is glucose molecule. Similarly, a wide array of different types of sugars can also enter glycolysis in different stages. Glycerol and fatty acids can also become a part of this pathway. So you can see here there is a communication between glycolysis and citric acid cycle and other catabolic pathways. Glycolysis can accept a wide range of carbohydrates for catabolism. So not only glucose but other forms of carbohydrates can also participate in the process of glycolysis. In the digestive tract, starch is hydrolyzed to glucose. Starch is broken down into its individual monomers, which is broken down in cells by glycolysis. Then once glucose is derived, glucose can be incorporated into glycolysis and then into citric acid cycle. Glycogen, which is the storage polysaccharide of animals and is stored in the liver, is present in the humans and many other animals, and it is stored in liver and muscle cells, and it can be broken down to glucose between meals as fuel for respiration. So it is the ultimate source of energy. The glycogen reserves in your liver, suppose you are on starvation for a whole day, then all your glycogen reserves can be used in a single day of starvation, and once the food is available in abundance, these reserves can be replenished again. Digestion of disaccharides, including sucrose, provide glucose and other monosaccharides as fuel for respiration. Proteins can also be used for fuel, but first they must be digested to their constituent amino acids. So the body is developed in such a way that it cannot derive energy from complex molecules. The molecules must first be broken down into their individual monomers, into their individual building blocks, and only then they will be able to serve as a source of energy, and only then they can become incorporated into metabolic pathways. Many of the amino acids are used by the organisms to build new proteins. Of course, amino acids are the building blocks of the proteins. So these amino acids are arranged in a polypeptide chain. The information for the synthesis of a polypeptide chain is of course present in the genes or the DNA. So the proteins, once perform their function, they are broken down by proteasomes, and the individual amino acids are released, which can be incorporated in the synthesis of some new proteins. Amino acids present in excess are converted by enzymes to intermediates of glycolysis and the citric acid cycle. So if the amino acids are in abundance, that is, if they are present beyond the need of the body, then those amino acids are broken down. The acetyl coenzyme A is formed, which is incorporated into Krebs cycle or the citric acid cycle. But before amino acids can feed into glycolysis or the citric acid cycle, their amino group must be removed, a process called deamination. So the amino group of the amino acids is removed through the process of deamination, and then they can be fed or incorporated into glycolysis and citric acid cycle. The nitrogenous waste is excreted from the animal in the form of ammonia, urea, or other waste products. Of course, these are the nitrogenous waste products. Ammonia is the most toxic, urea is the second, and then the third, in the third category comes other products. Catabolism can also harvest energy stored in fats obtained either from food or from fat cells in the body. So the human body and the body of the animals also has this ability of storing excess fats. So when the fats are destined to be used as the source of energy, these fats are either summoned from the fat reserves in the body or are taken up as food. After fats are digested to glycerol and fatty acids, the glycerol is converted to glyceraldehyde 3-phosphate, which is an intermediate of glycolysis. Recall the glycolytic pathway, where in the fifth step glyceraldehyde 3-phosphate and dihydroxyacetone is produced. So the glyceraldehyde 3-phosphate produced as a result of breaking down of fatty acids can directly enter from the fifth stage onwards. Most of the energy of a fat is stored in the fatty acids. A metabolic sequence called beta oxidation breaks the fatty acids down to 2 carbon fragments, which enter the citric acid cycle as acetyl coenzyme A. So the lipids can enter into the glycolytic pathway after glyceraldehyde 3-phosphate is formed from them, and they can also enter as acetyl coenzyme A into Krebs cycle. Now the NADH and FADH2 are also generated during beta oxidation. Remember, these are generated during glycolysis and Krebs cycle, and they are high energy electron carriers. So they can also be produced through beta oxidation of fatty acids. So this is yet another advantage of using fatty acids as a source of energy. they can enter the electron transport chain, leading to further ATP production. Now, fats makes excellent fuels in large part due to their chemical structure and the high energy levels of their electrons present in many carbon-hydrogen bonds equally shared between carbon and hydrogen compared to those of carbohydrates. So the carbohydrate glucose is a 6-carbon molecule, whereas the fats consist of 16 to 18 carbon molecules. So they provide more energy as compared to carbohydrates. A gram of fat oxidized by respiration produces more than twice as much ATP as a gram of carbohydrates. Why? Because glucose consists of 6 carbon molecules, fatty acids consist of 16 to 18 carbon molecules. So their breaking down provides more energy and more intermediates. Unfortunately, this also means that a person trying to lose weight must work hard to use up fat stored in the body, because so many calories are stockpiled in each gram of fat. So because the fat generates so much energy, and therefore if you want to burn the fats stored in your body, then you will have to undergo strenuous exercise. Biosynthesis in anabolic pathway, cell needs substances as well as energy. Not all the organic molecules of food are destined to be oxidized as fuel to make ATP. So all the organic molecules present in the body cannot be used to derive ATPs from. In addition to calories, food must also provide the carbon skeleton that cells require to make their own molecules. So basically the food we eat, the carbohydrates, the proteins, the fats, the lipids, when these molecules enter our bodies, they are broken down into their individual monomers, and then these monomers can be utilized by the body to synthesize other molecules. Some organic monomers obtained from digestion can be used directly. For example, as previously mentioned, amino acids from the hydrolysis of proteins in food can be incorporated into organisms' own proteins. So when you consume proteins inside your body, those proteins are broken down into their individual monomers, into individual building blocks called amino acids, and then these building blocks can be used by the body to make proteins of their choice. And from where the instructions will come for the synthesis of a specific protein? Of course, from DNA. So often, however, the body needs specific molecules that are not present as such in food. So sometimes your body requires some specific molecules which are not directly present in the food. So there must be some kind of biosynthetic mechanism through which your body can synthesize those molecules from their building blocks. Compounds formed as intermediates of glycolysis and the citric acid cycle can be diverted into anabolic pathways. Now remember what I said in the beginning, that glycolysis and citric acid cycle, they accept intermediates from other pathways, and the intermediates of glycolysis and Krebs cycle can become part of other pathways also. So the intermediates of glycolysis and citric acid cycle can be diverted into anabolic pathways, means they can come out from glycolysis and Krebs cycle and become part of any other pathway, and they can act as precursors. Precursors means the starting point of a molecule from which the cell can synthesize the molecules it requires. For example, humans take up half of the 20 amino acids in proteins by modifying compounds siphoned away from the citric acid cycle. The rest are essential amino acids that must be obtained in the diet. So remember, once I said in the class that some of the amino acids cannot be synthesized inside the body and they must be taken from the food. These amino acids are called essential amino acids, and other amino acids which can be synthesized in the body are called non-essential amino acids. Remember, this non-essential term does not refer to the importance of those amino acids. It refers to the fact that the body can synthesize them. Otherwise, the importance of essential and non-essential amino acids is equal. So from where these non-essential amino acids are produced, the intermediates of citric acid cycle can become precursors and enter other pathways, which leads to the synthesis of amino acids. Also, glucose can be made from pyruvate, and fatty acids can be synthesized from acetyl coenzyme A. So if energy is required, then fatty acids are broken down to acetyl coenzyme A through beta oxidation, and then acetyl coenzyme A enters into Krebs cycle, and if energy is in surplus amount, then acetyl coenzyme A can come out from Krebs cycle and enter a pathway which leads to the synthesis of fatty acids. Similarly, glucose, which is the starting point of glycolysis, is ultimately broken down into two molecules of pyruvic acid, and then this pyruvic acid enters liver, and through Cori cycle the glucose molecule is generated back from pyruvate. So depending upon the availability of the molecules and the need of the body. Of course, these anabolic or biosynthetic pathways do not generate ATP but instead consume ATP. So remember that the synthesis and the polymerization process requires ATPs, whereas when molecules are broken down, when catabolism occurs, then energy is liberated. In addition, glycolysis and the citric acid cycle function as metabolic interchanges that enable our cells to convert some kinds of molecules to others as we need them. So these two pathways are of primary importance because the metabolic products of other pathways can be incorporated into those pathways, and the intermediates of those pathways can be incorporated into other anabolic pathways. So it provides a perfect highway, a perfect metabolic highway. For example, an intermediate compound generated during glycolysis, which is dihydroxyacetone phosphate in step 5, can be converted to one of the major precursors of fats. And remember that once the fats are broken down, glycerol is formed, and then glycerol is converted to glyceraldehyde 3-phosphate, so it can also enter into glycolysis. If we eat more food than we need, we store fat even if our diet is fat free. So if the fats are in excess, then they are stored in the body. Metabolism is remarkably versatile and adaptable. So it's versatile means a lot of reactions and pathways are there inside a living system, and to a certain extent there is also interaction between those pathways, and those pathways have the ability to adapt to the circumstances, to adapt to the requirement of the cell. If energy is in surplus, then the energy is stored. If energy is in deficient amounts, then molecules are broken down. If amino acids needs to be synthesized, then acetyl coenzyme A enters that pathway. If energy is in deficient concentration, then amino acids are broken down and forms acetyl coenzyme A and enters into Krebs cycle. So depends upon the need of the hour. Now, one very important thing that occurs not only at the DNA level but also at the cellular level is regulation. I hope you remember the process of gene regulation, where the product of a gene is regulated. Similarly, on the cellular level, we also have a regulation where different metabolic pathways are regulated depending upon the need of the cell. How much products are required, for how long they are required, and when they are not required. So it all depends on the situation, on the circumstances inside the cell, and why there is the process of regulation. The process of regulation is important to maintain homeostasis. Basic principles of supply and demand regulate the metabolic economy. So when substances are required, when they are not required, the cell does not waste energy making more of a particular substance than it needs. So suppose you have 10,000 rupees, and with those 10,000 rupees you can buy food stuff, you can buy books, you can go and visit places, so you can carry out several activities using those 10,000 rupees. And suppose you utilized all those 10,000 rupees and popped up your mobile account, then obviously you will run short of resources, you will run short of money to use in other areas of your life. So similar is the case with the energy and those pathways where a product is only synthesized in specific amount which can meet the need of the cell. If that product is synthesized in surplus amount beyond the need of the cell, then obviously it will be a waste of the important resources. If there is a surplus of a certain amino acid, for example the anabolic pathway that synthesizes that amino acid from an intermediate of the citric acid cycle is switched off, We have already discussed this example where isoleucine synthesis takes place from threonine. So when threonine is in surplus amount, then this threonine itself attaches to the enzyme and it blocks the active site of the enzyme, so the enzyme is unable to synthesize further isoleucine. When the amount of isoleucine decreases in the body and the body is in need of synthesis of isoleucine, then this isoleucine is released from the allosteric site. As a result, this enzyme comes into its active functional form and it starts catalyzing threonine. So it all depends upon the need of the body, the need of the cell. And this is called feedback inhibition, when one of the intermediates of a pathway acts as a regulator on the same pathway. The most common mechanism for this control is feedback inhibition. The end product of the anabolic pathway inhibits the enzyme that catalyzes an early step of the pathway. So the end product is isoleucine. Now it inhibits the enzyme which catalyzes one of the early steps of the pathway. This prevents the needless diversion of key metabolic intermediates from uses that are more urgent. So the intermediates which can be incorporated in some other anabolic pathways are conserved, they are not wasted. The cell also controls its catabolism. If the cell is working hard and its ATP concentration begins to drop, cellular respiration is speed up. When there is plenty of ATP to meet demand, respiration slows down, sparing valuable organic molecules for other functions. Remember the third step of the glycolytic pathway, where if ATP is abundant, then ATP attaches to the allosteric site of the enzyme phosphofructokinase, which catalyzes the third step of the pathway. So why is this process occurring? Because when ATP is abundant, then there is no need to break down more glucose, because the end product of glucose is ATP synthesis, the synthesis of ATPs, and ATPs are already in surplus amount, so the glucose molecules must be conserved for times when the cell is running short of energy. Again, control is based mainly on regulating the activity of enzymes at strategic points in the catabolic pathway. As shown in figure on the right, one important switch is phosphofructokinase, the enzyme that catalyzes the third step of glycolysis. That is the first step that commits the substrate irreversibly to the glycolytic pathway. So remember that the products of first and second reaction of glycolysis can go back and form glucose again, but the third step is a very important step. It is a rate limiting step, and once the pathway crosses the third step, then it is committed to reach the end. So by controlling the rate of this step, the cell can speed up or slow down the entire catabolic process. Phosphofructokinase can thus be considered the pacemaker of cellular respiration. So if the energy is required, then phosphofructokinase attaches ATP to its active site and it catalyzes the process with even more efficiency, and when ATPs are in surplus amount, then ATP attaches to the allosteric site of the phosphofructokinase enzyme and the pathway stops. So here you can see the glucose molecule which is broken down. Fructose 6-phosphate is formed. Now this is the third step and it is catalyzed by phosphofructokinase. So when ATP is in abundant amount, then this enzyme is blocked and the pathway cannot move ahead. When ATP is in deficient amount, then ATP binds to the active site and this pathway moves ahead with even more efficiency. So it all depends upon the presence and the abundance of ATP molecules. It can inhibit the pathway by blocking to the allosteric site of phosphofructokinase, and it can also stimulate the pathway by binding to the active site of phosphofructokinase enzyme. Phosphofructokinase is an allosteric enzyme with receptor sites for specific inhibitors and activators. It is inhibited by ATP and stimulated by AMP, adenosine monophosphate, which the cell derives from ADP. So ADP has 2 phosphates and AMP has 1 phosphate. So obviously when ATP is in abundant amount, then it acts as an inhibitor. When AMP is in abundant amount, then it acts as an activator. As ATP accumulates, inhibition of the enzyme slows down glycolysis. The enzyme becomes active again as cellular work converts ATP to ADP. So when the level of ADP rises, then the enzyme is activated. When the level of ATP rises, then the activity of the enzyme is inhibited. Phosphofructokinase is also sensitive to citrate. Citrate is one of the intermediates of citric acid cycle. If citrate accumulates in mitochondria, some of it passes into the cytosol and inhibit phosphofructokinase. This mechanism helps synchronize the rate of glycolysis and the citric acid cycle. So this mechanism, this blocking of phosphofructokinase by the intermediate of citric acid cycle enables the two pathways to move at a proper pace, at a proper rate. As citrate accumulates, glycolysis slows down and the supply of pyruvate and thus acetyl groups to the citric acid cycle decreases. So citrate is one of the intermediates of Krebs cycle. So if the concentration, if the amount of citrate is higher, then this starting point of the pathway must be stopped to a point when all the citrate is converted into its intermediate forms, and then this pathway starts again. So when it accumulates, it stops the pathway, it stops the reactions coming before this point. If citrate consumption increases either because of a demand for more ATP or because anabolic pathways are draining off intermediates of the citric acid cycle, glycolysis accelerates and meets the demand. So because glycolysis provides raw material for Krebs cycle, so when the intermediates of the glycolytic pathway are incorporated into other pathways, then obviously there is a need to pacify this process so that intermediates can also be provided to the Krebs cycle. So then this process occurs with even more speed so that intermediates can be provided to other pathways as well as Krebs cycle. Metabolic balance is augmented by the control of enzymes that catalyze other key steps of glycolysis and the citric acid cycle. Cells are thrifty, expedient, and responsive in their metabolism. Review the first page of this chapter to put cellular respiration into the broader context of energy flow and chemical cycling in ecosystems. So the energy that keeps us alive is released, not produced, by cellular respiration. This is the last sentence of 5th chapter, and it quite beautifully summarizes this whole process, that the purpose of glycolysis, citric acid cycle, and electron transport chain is to release energy which is stored in the molecules, and energy is not produced. Remember, energy is already there, and these processes are only aimed to release energy. So this sentence validates the first law of thermodynamics, which says that energy can neither be created nor destroyed, but it can convert from one form to another form. So the energy in organic molecules is stored in the bonds, a kind of potential energy, and then this energy is derived and stored in another form of potential energy, where the third phosphate is attached with the ADP and ATP is formed. This was the end of the lecture. Thank you.